All right, so here in 14.3, we're now going to talk about the specific antibacterial drugs and then their mechanisms, how they work. So first, when we're talking about antimicrobial drugs, we kind of keep it general, the first portion of the chapter saying antimicrobial, but most of them are antibacterial. We will talk about other antimicrobials, but when we think about these things, we're mostly talking about antibacterial drugs, so against bacteria. The reason why most of our antimicrobial drugs are antibacterial is because they are prokaryotic, and their prokaryotic membranes provide a greater variety of targets for selective toxicity, meaning it is not very easy, but much easier for us to develop an antibacterial because they are prokaryotic versus eukaryotic, which our cells are. So if we are trying to develop an antimicrobial drug for something that is a eukaryote, like fungi or parasites, not viruses, but it can be very difficult because they have very similar cell structures to our own. So it's hard to kind of find a way to kill them without killing our own cells. Whereas bacteria are prokaryotes, there are lots of ways that we can kind of attack a prokaryote when it's not going to be hurting our cells, our eukaryotic cells. Our selective toxicity, so it selectively kills or inhibits the growth of our microbial targets. Doesn't cause much harm to us. That's what I was just saying. That's why most of them are antibacterial. It's easier. We can put a whole bunch of chemicals in our body that are going to kill their cells, but won't even touch our cells. Of course, then there are also some side effects and things, so sometimes they do. But then the mode of action. So the way the drug affects the microbes at a cellular level, again, is not going to affect our cells as much because we are eukaryotic organisms. So this is a general overview of the modes of action that we'll be talking about. Up in the right-hand corner here, I have a reminder of what our gram-positive bacterial cell walls look like and our gram-negative bacterial cell walls look like, because it's important that you recall and understand the differences between the gram-positive and the gram-negative. So recall that here's our cytoplasm, the interior of the cell. In a gram-positive bacteria, we have the plasma membrane here, and then we have this big, thick layer of peptidoglycan. And that's going to make up our bacterial cell wall. We have our plasma membrane and then the cell wall, which is the peptidoglycan. Recall that in our gram-negative bacteria, we also have this plasma membrane. However, then we have this periplasmic space here. And then we have our peptidoglycan cell wall, but remember it's very thin comparatively. But even though it's thin, what we then have on the outside of that, or more superficially, is this giant outer membrane. This giant outer membrane contains this lipoprotein or lipid A, our lipopolysaccharide. And then this gives our gram-negative bacteria kind of an advantage here. They have this whole other thick outer membrane, which is an additional plasma membrane with the addition of that lipopolysaccharide. So keep that in mind as we move forward and we talk about which types of modes of action are used on gram-positive versus gram-negative. It's also a good way as you are studying these different modes of action and these different antibacterials to remember which ones are going to affect either gram-positive or gram-negative because of their modes of action. So down here in the lower left area here, we have the different types of modes of action that we are going to talk about. So for example, we talk about our antibacterials that affect our cell wall. That's going to be its mode of action. It's going to somehow disrupt the cell wall, and we'll talk about the different ways that it does that. But you should know the different types of antibacterials for all of these different types of modes of action. So for the cell wall, we'll talk about the beta-lactams, including the penicillin, cephalosporins, monobactams, carbapenems, and then the glycopeptides, including vancomycin, and then bacitracin. So those are all going to be different types of antibacterials that are going to somehow affect the cell wall of a bacteria to stop it from dividing if it's bacteriostatic, or to kill it if it's bactericidal. We also have those antibacterials antibiotics that are going to affect the plasma membrane. So when we talk about the cell wall, we have the outer cell wall, and then we have the inner plasma membrane. So we'll talk about the different antibacterials that will affect the plasma membrane that will cause it to either stop growing or to kill it. Those that affect the ribosomes, recall that prokaryotic cells, bacteria, have different ribosomes than our eukaryotic cells. Recall that in chapter 3 when we studied that, that was important, and this is why it's important. Because since they have different ribosomes, then we can create antibacterials that are going to affect those specific ribosomes without affecting our own ribosomes. Also, we are able to affect the metabolic pathways, so we'll talk about these different types of pathways that we can affect for these bacteria. And then we can go in and we can affect the DNA synthesis or RNA synthesis. And again, this is because we've studied this and we know that there are differences between DNA synthesis in prokaryotic cells, meaning bacteria, and eukaryotic cells, meaning our own eukaryotic cells, and same thing with our RNA synthesis. So we can then develop these antibacterials to go in and attack these things at the places where we have differences between our prokaryotic bacteria and our eukaryotic cells within ourselves. So first, let's take a look at our inhibitors of cell wall biosynthesis. So our cell wall antibacterials. So the first group are the beta-lactams. The reason they're called that is because they have this beta-lactam ring. The prime example of that is penicillin. So penicillin is a beta-lactam. Our beta-lactams have this beta-lactam ring, and that's, you can see in the central area here of the molecule. But then we have this R group. This on its own here is going to be penicillin. But then remember that we have our semi-synthetics, and when we have our semi-synthetics, we can then take this R group and we can replace it with different things, which we'll see in a moment. So our beta-lactams have the beta-lactam ring, and what this does is it's going to block the cross-linking of peptide chains when the bacteria is making peptidoglycan. So remember, peptidoglycan is part of the cell wall. As the bacteria is making the cell wall, this beta-lactam ring is actually similar in structure to a subunit of peptidoglycan. And so it looks like a subunit of peptidoglycan. So the bacteria, particularly these penicillin binding proteins, or PBPs, actually grab onto the penicillin, for example, the beta-lactam, and it tries to incorporate that beta-lactam into the peptidoglycan, making peptidoglycan. Of course, it's not actually the subunit that it's thinking that it is. We've tricked it utilizing something that looks like it, this beta-lactam ring. What this then does is it disrupts the cell membrane. It doesn't attach the way it's supposed to, and then it ends up causing cell lysis. So this is bactericidal. So since the cell is not able to then make its peptidoglycan, make its cell wall, then it bursts and it dies. So we have our other beta-lactams. So for example, Penicillin G, Penicillin V, you can see these here, Penicillin G and V. These are natural antibiotics. They just come from fungi, so those are not semi-synthetic. They affect gram-positive pathogens and some gram-negative pathogens. Remember, that's because we have a whole lot of peptidoglycan in these gram-positive pathogens, not as much in the gram-negative, so it's not going to affect the gram-negative as much. Then we have our semi-synthetic penicillins, so we're changing that R group, and it's going to change some of the attributes. So you can see these are semi-synthetics here. They provide increased spectrum of activity, also stability, acid stability, and then resistance to penicillinases. So like methicillin, for example, does. So we're smart, but so are bacteria. So if we make a beta-lactam, for example, that's causing the cell wall to not be formed correctly and killing bacteria, the bacteria then are going to come up with a way to combat this, just like it would in the soil, for example. If one bacteria comes up with a way to kill another bacteria, that bacteria is going to try to find a way to defend itself. So then there are penicillinases that we have found in bacteria that are going to kill and get rid of penicillin so that it can go about its business making its peptidoglycan wall. However, things like methicillin are then resistant to that. So we, again, can adjust that R group to try to give it different effects. Continuing with the beta-lactams, we have our cephalosporins, which is a group of beta-lactams. They block transpeptidase activity of the penicillin binding proteins. So transpeptidase, ACE meaning an enzyme, and then trans meaning to transport or to move peptides. So these penicillin binding proteins, or these PBPs, remember they take this subunit, which we try to trick it with the beta-lactam ring, take the subunit and they move it to try to build their peptidoglycan cell wall. So cephalosporins also block these PBPs. So rather than tricking it and messing up the cell wall, instead what it does is it attaches to and it blocks these PBPs. But it also has increased resistance to beta-lactamases. Again, the bacteria, being smart, comes up with a way to try to thwart what we're doing. So beta-lactamases, ACE meaning an enzyme, this is an enzyme that is going to destroy our beta-lactams that we've come up with. So cephalosporins, however, have increased resistance to that. So we have our cephalosporin C, similar to penicillin, but can go against more gram-negative. Has two R groups. We can see it down here. There's an R group here and R group here. So it increases its diversity. Other beta-lactams are carbapenems and monobactams. So you can see those down here. Our monobactam has a difference. It has a sulfur here. And then we have our carbapenem, which is more of a broad spectrum semi-synthetic. So the next group of our inhibitors of cell wall biosynthesis are the glycopeptides. These, unlike our other ones previously, or not unlike all of them, I guess, are bactericidal. Back up. They're bactericidal. So some examples of our glycopeptides, or one example is vancomycin. You should know that. This is a natural antibiotic from an actinomycete. It's a very large complex molecule. What this does is it binds to the peptide chain of some of those cell wall precursors. So instead of tricking those PBPs and having the beta-lactam be attached on for peptidoglycan as a subunit and then it not working on the cell wall, our glycopeptides, what they do is they just glob onto a peptide chain of the cell wall of these precursors, these peptide chains, and then it just blocks the cell wall from being incorporated into the peptidoglycan. It blocks these subunits. So if it globs onto the subunit, that subunit can no longer be used to make peptidoglycan, therefore it can no longer be used to make the cell wall, and then the cell will die. It does also block transpeptidation, so grabbing those polypeptides and bringing them to build the glycopeptide, or peptidoglycan, sorry. It is bactericidal against gram-positive, again, because we've got all this peptidoglycan here. It's not active against our gram-negatives due to its inability to get past this large outer membrane. Remember, that's a barrier. Our last inhibitor of cell wall biosynthesis is bacitracin. Originally came from Bacillus subtilis. It blocks the activity of a specific cell membrane molecule. And this one is responsible for the movement of the peptidoglycan precursors, so these subunits, from the cell cytoplasm to the exterior of the cell. So what it does is it's going to block the process of these little tiny polypeptides moving from inside of the cytoplasm and getting them to the exterior of the cell to be incorporated into the cell wall or into the cell membrane. So rather than the pieces being attached to the peptidoglycans and it not being the right piece, instead of it attaching to the enzyme that's kind of moving these pieces around to build the peptidoglycan, instead, in this case, what it's going to do is prevent the movement of these peptidoglycan precursors from inside of the cell getting to the exterior of the cell. So then it can't be incorporated into the cell wall. It's effective against a wide range of bacteria. For example, against gram-positive that's found on the skin. It can be administered topically, orally, or intramuscularly. However, it can be damaging to the kidneys. So that is a risk. It is commonly combined with neomycin and polymyxin in things like Neosporin. That's why we see it topically. So down here at the bottom, we have our kind of summary of our mode of action where we inhibit cell wall biosynthesis. We can see that there are 3 different targets here. These are PBPs, the penicillin binding proteins. So if we're targeting the penicillin binding proteins, we're talking about our beta-lactams and those others listed there, the different drugs. If we're targeting our peptidoglycan subunits, then that would be our glycopeptides. And then if we're targeting the peptidoglycan subunit transport, moving from inside to the exterior of the cell, then that would be our bacitracin. So the second group, or the second mode that we're going to talk about, is protein biosynthesis. So again, remember that our cytoplasmic eukaryotic ribosomes, our ribosomes in our cells, are 80S. Remember the S is a kind of a weight. So 80S, which comprises of our 2 different subunits of 40S and 60S. No, they don't add up to make 80S, but that's the way this is. So these are different than bacterial ribosomes, which recall are 70S. And with 70S, we actually have 30S and 50S. And again, the reason that it's important that you understand this is because note that none of our ribosomal subunits match. We don't have a 30 or a 50, they don't have a 40 or a 60, so this makes it a good target for our antibacterial drugs because they're different, so we can target theirs and not hurt ours. So the first group of our protein biosynthesis inhibitors are aminoglycosides. So these are large, they're highly polar, and what they do is they attach to the 30S subunit, so the small subunit. When this happens, it's going to impair the proofreading ability of the ribosomal subunit. So remember, the ribosomes are going to be making those proteins. So what happens is it mismatches the codon and the anticodon. So it globs onto that small subunit. It kind of messes up the small subunit, so it doesn't match the codon with the anticodons correctly. So then what happens is then we have our incorrect amino acids are going to be added to the proteins, and that's going to shorten the proteins. And then when they get incorporated into the cell membrane, then the cell membrane is not correct. We don't have the correct proteins, or they don't have the correct proteins in the cell membrane, and then it bursts. It will kill the cell. So these are broad spectrum antibacterials because we're talking about the proteins that are being made for the cell membrane, and both gram-positive and gram-negative have cell membranes, so it's broad spectrum. However, they can be nephrotoxic, so toxic to the kidneys, neurotoxic, so toxic to the nervous system, and ototoxic, so toxic to our ears. So there's some big risks with these. Some examples are streptomycin, gentamicin, neomycin, and kanamycin. So those are aminoglycosides. Other inhibitors of protein biosynthesis are tetracyclines. So our tetracyclines are going to also bind to that 30S subunit, the small subunit in the bacteria. What it does here is that these are going to block the association of that tRNA. Remember, the tRNA is going to be bringing that individual amino acid to attach it to the growing polypeptide chain. So it blocks the association of the tRNA with the ribosome during translation. So this is actually bacteriostatic. So it's not going to kill it anyway. It's not popping the cell wall, or it's not breaking the cell membrane to burst it. Instead, what it's doing is it's just making it very ineffective. So it's attaching to that small subunit. It's making it so that that tRNA can't match up correctly, and then protein synthesis isn't really going to be happening. It's naturally produced by streptomyces. However, we have semi-synthetics, so doxycycline and tigecycline are some examples. We have some side effects. Phototoxicity, so sunburn with light exposure. So sometimes if somebody is given one of the tetracyclines, it's told that they shouldn't spend large amounts of time out in the sun because there is the risk of phototoxicity, so you can get sunburn very quickly. There's also the risk of permanent discoloration of developing teeth, so giving tetracycline to a small child is not ideal because it can discolor their teeth. Also, liver toxicity with high doses, or of course in patients that have some sort of impairment. So kidney impairment because they're not eliminating it well enough, and if they're not eliminating it via the kidneys, it builds up in the liver and can become toxic to the liver. So down here we have our major classes of protein synthesis inhibiting antibacterials, so those that are inhibiting the actual production of proteins. We talked about the aminoglycosides and the tetracyclines, and then we have chloramphenicol and the macrolides and lincosamides. You can see that in this case, our macrolides are going to bind to the 50S ribosomal subunit, so that's the larger subunit. And then what they're going to do is they're going to prevent that peptide bond from forming, and so it's stopping that synthesis. So our macrolides, for example, they are also broad spectrum. They are bacteriostatic, and that's because they block that elongation of proteins. What they do is they inhibit the peptide bond from forming. So we're not attaching to that small subunit. If you look back at the last slide here, so if we're not forming that bond, we're not making the proteins. So again, that's why it's bacteriostatic. Erythromycin is an example of macrolide. Our macrolide, azithromycin, so it's broader spectrum, fewer side effects, lasts longer. This is what we see in our Z-pac is azithromycin that's often prescribed. And telithromycin, so this has an increased potency and activity against our macrolide-resistant pathogens, so those that have become resistant. So for example, in community-acquired pneumonia. There is some serious liver issues here though. Hepatotoxicity is toxicity for the liver. This one has the strongest black box warning label. So black box meaning the big black box that you get when you get all that paperwork related to one of the pharmaceutical drugs that you get, because it can have some serious issues with the liver. So taking lots of Z-Paks is not a good idea. However, when it's necessary, it can be very beneficial. So as we mentioned, binding to the 50S subunit and interfering with peptide formation. Our lincosamides are another example. Chloramphenicol, another example. So the lincosamides are effective against strep and staph. Chloramphenicol is seriously dangerous to humans. There are lots of side effects, so it's not used very often. We do see this in veterinarian medicine. It doesn't affect them as much as it affects us. This was, however, the first FDA-approved broad-spectrum antibiotic. So way back when, we were like, this is the newest, greatest thing, and then we realized there are a lot of side effects, but by that time we were making other things. Oxazolinidones are a new class of broad-spectrum synthetics that are based on this same type of thing where they bind to the 50S subunit, and then they're going to interfere with the formation of the protein. In this case, they're interfering with the initiation complex, so that very first tRNA that comes along with the very first amino acid. So this is just, again, a summary of our protein biosynthesis inhibitors. So you should know each of these. You should know how they work. You should know examples of the particular drugs as well. So the next one are inhibitors of membrane function. The first one are polymyxins. These are natural polypeptide antibiotics. What they do is they interact with the lipopolysaccharide component of that outer membrane and gram-negatives. So they disrupt both of the plasma membranes, and then of course they have cell lysis. So these are not selective. They can target and damage kidney cells and nervous system cells if they are administered systemically. So if they're put into the system, they can actually damage our cells as well. So although it is a natural polypeptide antibiotic, it can also affect our cells. Even though it is going to interact with the lipopolysaccharide, which recall we don't have lipopolysaccharides in our plasma membranes, it also disrupts the membrane part, and so it can cause problems for us. One example, or some examples of the polymyxins, are polymyxin B, which we see in Neosporin, polymyxin E, which is colistin, It's used to decontaminate the colon for surgery or in people that are immunocompromised. Sometimes people have to go in and have surgery on the colon, and when they do that, if a person or if the person goes in and has surgery on the colon and they cut the colon open with all of the bacteria that's in the colon, it is possible for that to cause problems with bacterial infections in the rest of the body if any of that bacteria were to reach the rest of the body. So sometimes they will decontaminate the colon, so they will actually wipe out all of the bacteria in the colon so that they can safely do surgery, or in immunocompromised if they are having a problem with some sort of pathogenic bacteria. Rather than trying to go with some narrow spectrum or other broad spectrum, they'll go in and they'll just wipe everything out. Then they need to start seeding the colon again, which is talked about in another section, which is when they'll start to do fecal pills, which is where people can then start taking pills that are filled with other people's fecal matter to get bacteria back in their body. Maybe not ideal, however, if a person is immunocompromised and they may die from bacterial infection, maybe it's better to go in and wipe out everything and then have to take some pills that might not be ideal rather than actually dying. Lastly, we have daptomycin. This targets gram-positive bacteria. It's administered intravenously. It has reversible toxicity in skeletal muscle, so it can be toxic, but it can actually be reversed. All right, so moving into our inhibitors of nucleic acid synthesis. So another mode is rifampin. So this actually blocks the RNA polymerase in bacteria. So remember that the RNA polymerase in bacteria is different than the RNA polymerase in our eukaryotic cells. So this is why we talk so much about those enzymes when we were going through that chapter as well. Its primary use is for Mycobacterium tuberculosis, and it's typically mixed with others. Some disadvantages of rifampin is that it increases the metabolism of other drugs, so it has an antagonistic effect, which can cause problems, and it can lead to hepatotoxicity and decreases the effects of the other drugs. So oftentimes this is used for Mycobacterium tuberculosis. However, if the person is taking something else, they have to seriously look at the differences and whether or not it is necessary to use rifampin. Next, we have our quinolones, also inhibitors of nucleic acid synthesis. Examples are Nalidixic acid, so this inhibits the activity of bacterial DNA Gyrase. So this blocks DNA replication, right? So we're not talking about RNA here, we're talking about DNA, nucleic acid. If we're going in and we don't have our DNA Gyrase, then we're not going to have DNA because we can't attach the pieces of DNA correctly. We have our fluoroquinolones. And recall that DNA Gyrase here is different than the enzymes that we use in our body. So again, we're attacking where they have differences with our cells. All right, so the fluoroquinolones they work by chemical modification to the quinolone backbone. This also blocks DNA Gyrase, so also attacking DNA Gyrase in this case. Examples are ciprofloxacin and levofloxacin. These are broad spectrum, so gram-positive or gram-negative. You probably have heard of ciprofloxacin, so it's among the most common antibiotics prescribed for urinary tract infections, for example, respiratory infections, abdominal issues, and also some skin infections. However, there are side effects. For example, phototoxicity, so remember not to be out in the sun. You can get sunburned very easily. Including neurotoxicity. There are actually lots of cases of neurotoxicity where a person actually has so much neurotoxicity that they have some serious pain throughout the rest of their lives and sometimes have inability to use their legs and things. Also cardiotoxicity. Some glucose, cardiotoxicity meaning toxic to cardio, meaning the heart. Glucose metabolism dysfunction, so an inability to break down glucose effectively. And then also there's an increased risk for tendon rupture, so actually breaking the tendons. So again, can be very, very helpful and is most commonly used as this ciprofloxacin, but there are lots of side effects that go along with it. So next we have our inhibitors of metabolic pathways. So metabolic meaning our building up of things, so metabolizing things, making things inside of the bacteria. So we're going to look at our anti-metabolites. In this case, what they're doing is they're competing with bacterial metabolic enzymes. So these are competitive inhibitors. So they are going to compete with molecules that bacteria are trying to use, and they're going to inhibit the pathway. So our sulfa drugs, our sulfonamides, are structural analogs, meaning they look very similar to an intermediate in folic acid synthesis. So since they look like this particular intermediate in folic acid synthesis, the bacteria is trying to make folic acid. What it does instead of using the molecule that it's supposed to use, it uses the sulfa drug. When it uses the sulfa drug, it's going to block folic acid synthesis, but not only does it block folic acid synthesis, that same process is used to make pyrimidines and purines for nucleotides. So not only is it going to stop folic acid synthesis, which is necessary for the cell, but it's also going to stop it making those nucleotides for DNA synthesis. So this is going to provide bacteriostatic inhibition, so it doesn't actually kill the cell, but what it does is it sure slows it down a lot because we're putting this in there and then it's thinking that it's using the molecules that it needs, and instead it's using the molecule we just gave it, which makes it not make folic acid or the nucleotides. However, it is selectively toxic to bacteria, meaning that it is toxic to bacteria. However, it can also harm humans. But we ingest folic acid, so we can still get folic acid, even though it can damage our process of making folic acid. However, allergies are very common with sulfa drugs. So if we look at the image over here on the right-hand side, this is the process that the bacteria wants to go through and normally goes through to make the nucleotides, and then also a step in here to make folic acid. So it utilizes this PABA with this enzyme, say this first enzyme. It's going to do its thing. The enzyme is going to attach the PABA with some other molecule and then make this dihydrofolic acid, this molecule here. Then that dihydrofolic acid goes to the second enzyme. That's why it's shaped different, to show that it's a different enzyme. And then it's going to do its thing and make tetrahydrofolic acid. And then that's going to go on to make a nucleotide, or a nucleoside, because it's the pyrimidine and purine part. When we put in our sulfonamide, our sulfa drug, this looks, you can see it looks very similar to the PABA. Since it looks so similar, it fits very nicely with this enzyme, this same enzyme here. And when that does that, we are not going to get dihydrofolic acid and then not going to get the rest of this process. So it's a structural analog, meaning it looks very similar structurally, and so it competitively inhibits this synthesis of the dihydrofolic acid. So it attaches because it looks the same, and then it competes. So if we put a whole bunch of the sulfa drug in there, yes, the bacteria will likely still grab some of the PABA and make some folic acid or make some nucleotides. However, if we inundate it with the sulfa drug, it's more likely to grab onto the sulfonamide and then use that, and then it stops that synthesis. So that's why it's bacteriostatic. Another type of metabolic pathway inhibitor is trimethoprim. So it is a structural analog of another intermediate in folic acid synthesis. So this is also bacteriostatic. So if we look all the way to the right here, again, this is a reminder of the process that the bacteria wants to go through using PABA, etc., as we just saw. In this case, when we're talking about trimethoprim, here, the bacteria is still able to use the PABA, so we're not affecting that at all. It goes through, utilizes this enzyme, and then what happens is that instead of having a lot of this dihydrofolic acid around, it will still have dihydrofolic acid around, but instead, if we inundate it with trimethoprim, trimethoprim is a structural analog of the intermediate here. So then when we're talking about the second enzyme here, then it's going to competitively inhibit this second enzyme. So then we're going to not get the tetrahydrofolic acid that then can continue on to make folic acid and the nucleotides. So oftentimes it's going to be used synergistically, right? So if we put in a sulfa drug and we put in trimethoprim, then we're blocking both steps in this pathway, and so that's going to increase the ability of these drugs, right? So we said that sometimes we have this synergistic reaction where we have one bacteriostatic drug and another bacteriostatic drug, and then both of those working together can be bactericidal. And that's what we see in this case, because if we put enough sulfonamide in there, enough trimethoprim in there, then we can basically completely block this process of making folic acid and the nucleotides. And if it can't make nucleotides, it can't make more bacteria and it will die. And it can't, if it doesn't have folic acid, it will die. So then it's bactericidal. So we see this in UTIs. These are used for UTIs, for ear infections, and for bronchitis. However, there has to be careful consideration of taking it during pregnancy because we're blocking that folic acid pathway. So again, remember, it's selectively toxic. So although a woman, when they're pregnant, oftentimes takes folic acid, we still would want to be very careful with taking these because it blocks that folic acid pathway. So our next mode of action is the inhibitors of ATP synthase. So our betaquiline, it interferes with the production of ATP synthase. So if we can't make, or if it's decreasing ATP synthase production, then we're going to be reducing ATP synthesis. If we're reducing ATP synthesis, then we're decreasing the energy for the cell. If we're decreasing the energy, then it no longer lives. We use this specifically for mycobacteria. Some side effects are hepatotoxicity. A lot of these are hepatotoxicity because our liver is where we go through and we make things that are toxic not toxic anymore, so we can get rid of them. So as all of these toxic things are going to the liver, it damages the liver over time. It also can cause a potentially lethal heart arrhythmia, so quite dangerous there. It is oftentimes reserved for otherwise untreatable tuberculosis cases, so mycobacteria tuberculosis. So again, because we have this heart arrhythmia problem and hepatotoxicity, it's reserved for severe cases. So that summarizes our modes of action, or this here is the summary of modes of action that we just went through. We talked about cell wall inhibitors, plasma membrane, talked about the ribosomes attaching to the 30S subunit, the 50S subunit. We talked about affecting those metabolic pathways, and then we talked about DNA synthesis and RNA synthesis, those that are going to affect all of those different areas and why they affect the different areas. So you should know everything that we just talked about. You should know all these different modes of action. You should know each of these types of antibiotics underneath there. You should know which ones work in what ways, and what their spectrum is. So if they're gram positive, gram negative, if they're broad spectrum, narrow spectrum, the information that's been given.